Welcome to the Explainer. Okay, let's just dive right into this microscopic city that literally makes up the fundamental unit of, well, you, the cell, right? I mean, every living thing, from a tiny bacterium to a giant redwood tree, and yeah, even you watching this, is built from these incredibly hardworking little structures. So, we're treating this as your definitive exam masterclass. I'm going to break the cell down into a super relatable factory analogy and point out those guaranteed high-yield exam takeaways. If you see a star icon pop up today, pay attention. That means it's a critical concept you absolutely need to know.
So, here's the game plan for our masterclass today. We've got 1. Cell basics and theory. 2. Boundaries and transport. 3. The cellular assembly line. 4. Control and power. And finally, 5. The big comparisons.
Okay, but before we get into how the factory works, we kind of need to talk about how we even found it in the first place. Think about this for a second. The absolute limit of resolution for the human eye is just 0.1 millimeters. If two dots are closer together than that, they just blur into one. Since a typical cell is way beyond our naked eye's limit, how on earth did we discover them?
Well, the answer is lenses. And this whole timeline brilliantly illustrates the foundation of all biology, the classical cell theory. It all kicked off back in 1665 when Robert Hooke used a self-designed microscope to look at some cork. He saw these little box-like compartments, and well, he named them cells. Fast forward a bit, and scientists Schleiden and Schwann figured out that actually all plants and animals are made of these cells. Then the real mic drop moment. In 1855, Rudolf Virchow unified the whole thing by proving that every single living cell arises only from pre-existing cells. No spontaneous generation here.
All right, let's move right into Section 1, Boundaries and Transport, or, as I like to call them, the gates.
So take a look at the fluid mosaic model. This is a massive exam topic, by the way. It shows the plasma membrane, which is a universal feature of literally all cells. It is incredibly thin, like we're talking 7 to 10 nanometers thin, and it's made of this lipid bilayer. You see those little blue shapes? Those are embedded proteins. Together, they act as this highly sophisticated, selectively permeable security gate. They let the good stuff in while perfectly blocking the bad stuff.
Now pay super close attention here, because this next concept is heavily tested. I mean, it's everywhere in biology exams. Osmosis. You already know diffusion is just particles moving from a high concentration to a low one, right? But osmosis is specifically the diffusion of water across that selectively permeable membrane gate we literally just looked at. The water always moves from where it's dilute, meaning there's more water, to where it's highly concentrated with solutes.
And because osmosis is such a big deal, you absolutely must memorize this table for your exams. It breaks down 3 distinct environments a cell can find itself in. First, isotonic. That just means the concentration is exactly equal inside and out. Then there's hypotonic. This is where the outside fluid is more dilute, so water actually rushes into the cell. And finally, hypertonic, where the outside is super concentrated, pulling water out of the cell.
This image brilliantly illustrates osmosis in action. On the left, those red blood cells are perfectly happy in an isotonic solution. Nice and balanced. But look at the right side. When you drop them into a hypotonic solution, water just rushes into the cells, blowing them up like little balloons. And get this, if too much water goes in, an animal cell can literally pop. It just bursts.
But wait a second, what about plant cells? Well, unlike animals, plants have this incredible extra layer of armor, a rigid cell wall made of cellulose. Mark this down, it's a crucial exam distinction. When a plant cell takes in a ton of water during osmosis, it swells up against this wall, but the wall actually pushes back. This is what keeps the plant firm and upright, and it completely prevents the cell from bursting. Pretty smart, right?
Okay, moving on to Section 2, the cellular assembly line. Let's step inside the cytoplasm.
First up on the factory floor is the endoplasmic reticulum, or ER for short. This is our main manufacturing area, and you definitely need to know the 2 types for your exams. The rough ER is covered in these tiny little ribosomes, which gives it a bumpy, rough look under a microscope. This is where proteins are synthesized. Then you have the smooth ER. No ribosomes here, hence smooth, and its job is all about making lipids, or fats.
Next, we have to meet the cellular post office. And this guy here is Camillo Golgi. He actually discovered the Golgi apparatus way back in 1898 while he was looking at the nerve cells of a barn owl. Crazy, right? The funny thing is, early microscopes were so weak that a lot of the scientific community straight up doubted his discovery for decades. It wasn't until the electron microscope came along that they finally proved him right.
So what you really need to remember for the test is this step-by-step connection between these organelles. Think of it like this. The ER acts as the assembly line, manufacturing those proteins and lipids. Then, those raw products get sent over to the Golgi apparatus. The Golgi modifies them, packs them up into little delivery boxes called vesicles, and dispatches them off to their final targets, whether that's inside or outside the cell.
Now, with all of this intense manufacturing and shipping going on, things are definitely going to get a bit messy. So you might be wondering, how exactly does the cell handle all the inevitable trash, the damaged parts, and the waste materials? How does it avoid turning into a microscopic landfill?
Well, the answer is lysosomes. These guys are the cell's dedicated cleanup crew. They're basically these little sacs filled with incredibly powerful digestive enzymes that are capable of breaking down unwanted proteins, fats, and even old, worn-out organelles. Definitely highlight this one with a huge star for your test. Lysosomes are absolutely essential for cellular waste management.
All right, Section 3, Control and Power. Let's talk about the nucleus and mitochondria. Okay, so the nucleus is the ultimate control room. It's wrapped in a double-layered membrane that has these tiny pores to strictly control what comes in and what goes out. Inside, it guards the master blueprints, your chromosomes, which are made of DNA. These direct literally all cellular activities. It even houses this little area called the nucleolus, which is basically a mini factory dedicated just to making ribosomes.
And here is another absolute exam favorite, the mitochondria. Yes, the powerhouse of the cell. It also features a double membrane, but check this out. The inner membrane is folded up into these finger-like projections called cristae. This is a neat trick to massively increase the surface area. Right here is where molecules get broken down to generate ATP, adenosine triphosphate, which is literally the universal energy currency the cell spends to do, well, all of its work.
Okay, we've made it to Section 4, the big comparisons. Let's look at some key cellular differences.
Now listen up, because teachers absolutely love asking you to contrast these specific concepts. First up, prokaryotic versus eukaryotic cells. You've got to know that primitive prokaryotes, like bacteria, are tiny and entirely lack a well-defined membrane-enclosed nucleus. Their DNA just kind of hangs out in a region called the nucleoid. Eukaryotic cells, on the other hand, are much larger, way more complex, and feature true membrane-bound nuclei and organelles.
Similarly, when you're looking at plant versus animal cells, you have to remember this core difference. Plant cells are defined by having those rigid exterior cell walls, massive storage vacuoles, and special organelles called plastids. Chloroplasts used for photosynthesis? Yep, those are plastids. Definitely star vacuoles and plastids in your notes because they are incredibly high yield for exams. Animal cells, meanwhile, completely lack cell walls and plastids, but they do feature those waste-destroying lysosomes we just talked about.
Finally, our cellular factory has to expand, right? Which brings us to cell division. Mitosis is your standard growth and repair cycle. It takes one parent cell and creates two exact genetically identical clones. Meiosis, however, is strictly for reproduction. It's a two-step process that results in 4 unique daughter cells, and here's the kicker. Each of them has exactly half the number of chromosomes as the parent. So we've mastered the foundational building blocks today, from the lipid gates all the way to the mitochondrial powerhouses. But honestly, it leaves us with an incredible mystery. Think about it. If you started as one single cell, dividing by mitosis to make identical clones, how do those tiny, perfectly identical factories eventually specialize to become your brain cells, your muscle tissue, and your bones? Keep that question in back of your mind as you continue your biology journey. Thanks so much for joining me on this explainer, and I will see you next time.